So I will be presenting now on the Fish and Wildlife Service's AIS surveillance that we conduct annually in Lake Huron. And I want to frame this up, the importance of this monitoring, the surveillance that we conduct. This does not take place in just Lake Huron that I'll be presenting on today, but it takes place in all of the Great Lakes. And so we're out there trying to effectively find a new invasive species before it becomes established, before it becomes problematic. And the earlier that we can really detect these species, should they become present in the lakes, the earlier we can provide that information to the lake managers, and they can take action to potentially eradicate or control invasive species before they become a big problem in the Great Lakes. Because we all know that once, as we see with brown goby, some of the dry-scented mussels, once they become established, they have some pretty dramatic impacts on the ecosystem. So our goal is to get out there and detect them as early as we can and provide that information to the lake managers. And I want to thank the Lake Huron Committee, of course, for allowing me the opportunity to present this information to everybody today. So I'd like to frame it up. There's 3 components to the invasive species program. 2 of them take place in Lake Huron, and then we actually have a 3rd component down in Lake Erie, the grass carp program, which functions under the Grass Carp Advisory Committee. I won't be touching on that today, but I will be talking about first our early detection and monitoring program. And so last year in 2023, we covered 8 locations from the St. Mary's River all the way down to Saginaw Bay. And these are high priority locations that have been identified as a highest risk for invasive species to become established there or to invade there should they enter Lake Huron. We 376 sites and units of effort. We identified 48,000 fish this last year across 83 different species, and we're happy to report that we had no new invasive species detections, but we did, however, have 2 potential range expansions, which I will get into here in a moment. So the 2 range expansions that we found this last year, the first being tube-nosed goby, and that was found just off of a Grand Lake outlet in northern Lake Huron. This is right outside of Presque Isle. And although we have not found tube-nosed goby in that location before, it's likely that they've been there for a while because they are present north and south of that region along the shoreline. So this might not be as significant as what we found with the Eurasian rough expansion that Lisa touched on in the St. Mary's River. And so historically, rough have been captured in Lake Superior and the upper St. Mary's River. And as you can see, they've been well established in Lake Superior for some time now since they invaded over here in the St. Louis River estuary back in the 1980s. However, we don't really have many populations of rough outside of Lake Superior. We did in 1996, we had a population pop up in Alpena, Thunder Bay. However, by 2003, the last rough was captured there in Thunder Bay, and we have not recorded any rough in Lake Huron since. We have had, well, I take that back. We have had some individual rogue captures in 2008 and 2011 in northern Lake Huron, but follow-up sampling has, we haven't found any other individuals, so we do believe that those were rogue individuals. And then in 2017, like Lisa mentioned, we did have 1 individual rough that was captured in the lower St. Mary's River, but since then we haven't turned up any rough in the lower St. Mary's River until this year. So this year, it was first DFO that captured rough in Lake George, which is up here in the lower St. Mary's River. And then in October, the Sault Ste. Marie tribe of Chippewa Indians collected an additional rough in the lower St. Mary's River up here. And so it's really critically important that we have such great partnerships with all the agencies that work up in the St. Mary's River for this AIS early detection. Because if it wasn't for these captures from our partners and the communication that we have with them, we wouldn't have been able to conduct any of the follow-up sampling that we did, which occurred at the end of October, right at the tail end of our field season, where we went back up to Lake George with our bottom trawl and we turned up 9 additional rough. So this is pretty significant because this is the first time that we're actually finding rough in quantity in the lower St. Mary's River. They do seem to be limited to Lake George right now. And moving forward, we do plan to conduct, really ramp up our bottom trawl and our sampling efforts in the lower St. Mary's River this year to determine what the extent of this population is and really track where that invasion front is potentially over the course of this next year. I do want to point out that as these detections were recorded, or as this information did come to light, this information was recorded out to the proper channels, the Lake Huron Committee, the Lake Huron Technical Committee, the St. Mary's River Fisheries Task Group, and Michigan DNR. So we will continue to make this information available to all the appropriate lake managers as we come across potential more rough this coming year. We also conduct some early detection and monitoring benthic invertebrate sampling. We only have one location in Lake Huron, and this takes place in Saginaw Bay. This last year, we had some sweep nets and some rock bags, and we essentially look for amphipods, gastropods, bivalves, and crayfish in all of our invertebrate samples. I don't have the information to present right now. We're actually outsourcing the IDs for all of those specimens that are in those samples, and we're pretty excited to get really detailed taxonomic IDs on all of those individuals and start building up a pretty robust data set and start tracking some of these species as far as the invertebrates go through time. The next part that I'll talk about is our invasive carp eDNA program. We do have sites down in Lake Erie, and we have a site up in Lake Huron. And this takes place in Saginaw Bay and Saginaw River. Last year, we sampled 440 water samples in early to mid-May, and we had 0 positive detections, which means that there was no presence of bighead or silver carp eDNA present in the Saginaw River. Of course, I'm sure that if we had positive detections, you probably would have heard about it by now. Moving into 2024, we will continue to do our annual monitoring of bighead and silver carp in Lake Huron. And this is just an example of what our sampling looks like. So we try and get as much spatial coverage as we can along the river. So we sample all the way from the mouth of the river to the confluence of the Shiawassee and Tittabawassee, and actually this next year we'll be moving even further upriver. So we're really just trying to get as much spatial coverage of the river as we can to look for eDNA for a bighead and silver carp. I'm going to leave this up here for a second. So this is a QR code that links to the Fish and Wildlife Service's public eDNA dashboard. And for those that are interested, you can access this dashboard through your phone. You just take a picture of it, and this will show all of the eDNA samples that have been taken by the Fish and Wildlife Service throughout the entire Midwest region, all of the Great Lakes, the Mississippi River Basin, and it has a lot of detailed information on there. It's really a really cool tool for folks to be able to go back and look at all where the positive detections have taken place, where negative detections have taken place, the amount of samples that we've taken over the last, I think going on 11 years now. And I guess while I have this up here, I'll make a plug for one of the new tools that Fish and Wildlife Service is developing, which is called metabarcoding. And this is a form of environmental DNA sampling. So for folks that aren't aware of what metabarcoding is, as it relates to eDNA, traditional environmental DNA sampling, you are looking at for targeted species. So for what we do, we're looking for targeted species being bighead and silver carp. And what we do is we go out and we take water samples. And those water samples, the geneticists will run their magic on it, and they'll tell us whether there's bighead or silver carp eDNA present within that water sample, which then kind of guides any follow-up actions. With metabarcoding, which is a new tool that we're starting to bring online in the next couple of years and we're starting to develop, we take a water sample and it doesn't just tell us 1 or 2 species that we're specifically looking for, but it will tell us the entire fish community that's present. So it could be an extremely powerful tool, even beyond AIS and invasive species detections. We'll be able to find T&E species, things that the states might be concerned about, and really just get a better idea of what the fish community looks like in an area, if they might not be vulnerable to some of the gear that we're deploying. And then I'm going to make one more plug here, and I'm actually going to ask the Invasive Species Fish and Wildlife Service staff to raise their hands. So Sharon, Kristen, Greg, Mike Sider, Jessica Shara. I'm actually going to have Scott and Susan raise their hands as well. So if anybody comes across any invasive species or any fish that they're concerned about that they maybe have trouble identifying, please reach out to myself. My information's up here, or any of those folks in the room that just raised their hand. We do have the expertise on staff to identify not just the fish that are present in the Great Lakes, but fish that are not present in the Great Lakes, and that might potentially be some pretty catastrophic invaders. So if we go out there and we're conducting our surveillance throughout Lake Huron, there's a lot more work that's being done on the landscape, and if anybody comes across anything that they're concerned about, or maybe they're just not sure if it should be there or not, we really encourage you to reach out to our experts, and we'll kind of help guide you through identifying what that specimen is, the communication to the appropriate lake managers, and then the significance of maybe what that capture is. And with that, that's my last slide, so I'll take any questions that you might have.